Welcome to RPB's Lunch and Learn, Eye on AMD. Research to Prevent Blindness is the leading nonprofit organization supporting research directed at the prevention, treatment, or eradication of all diseases that damage and destroy sight. RPB was founded in 1960, and over our 60-year history, we have awarded more than $383 million in research grants to the most talented vision scientists at the nation's leading medical schools. As a result, RPB has been associated with nearly every major breakthrough in the understanding and treatment of vision loss. In today's educational session, our second of 4 Lunch and Learns, you will hear from 2 RPB-supported talented scientists about age-related macular degeneration, or AMD, pathology, disease management, and new research directions. Both of our speakers received the RPB Catalyst Award for innovative research approaches for AMD, which support completely novel research approaches to AMD with the ultimate goal of creating new and effective therapies. We are fortunate to have 2 excellent funding partners for these awards, the American Macular Degeneration Foundation and Dr. H. James and Carol Free. We are thankful to AMDF and the Free family for making these awards and this research possible. Thanks also to all the generous supporters of RPB who make our work possible. The importance of supporting medical research has never been so apparent. We thank you for your partnership in supporting innovative vision research to ensure that new breakthroughs are possible. But getting back to today's session, let me introduce our moderator. Carmelo, or Carl Romano, PhD, is currently the head of ophthalmology research at Regeneron. Previously, he directed the Retina Discovery Research Program at Alcon Labs. Prior to his career in pharma, he had a 25-year academic career, most of it spent at Washington University Medical School in St. Louis, in the ophthalmology and neurobiology departments there. His training included an undergraduate degree in biochemistry from SUNY Stony Brook, a PhD in pharmacology at Stanford, as well as postdoc positions and junior faculty appointments at Yale, Rockefeller University, and the University of Pennsylvania, where he specialized in neuropharmacology. RPB is proud to have supported Dr. Romano with the prestigious RPB Lou R. Wasserman Merit Award during his academic research career. Dr. Romano will introduce our 2 speakers for today and moderate the Q&A session at the end. Each speaker will speak for 20 minutes and I will serve as timekeeper. I will give each speaker a warning at the 18-minute mark. We want to make sure that we have time at the end for a robust question and answer session. Dr. Romano, take it away.
Thank you very much, Brian. It's a pleasure to be here. It's a pleasure to be here. RPB supported me, and I've gotten numerous grants when I was in university setting from them, and it's a pleasure to be able to participate in this very nice and important proceedings today. So let me just give a little introduction to the topic. Until about 10 years ago, if you received a diagnosis of exudative AMD, this was in effect a death sentence for your eyesight. Within a couple of years, you will have lost most, if not all, of your visual acuity. The advent of anti-VEGF therapies changed all of that. Now, most people diagnosed with AMD regain much of their lost vision, and in virtually all patients, the progression of the disease is halted. This is miraculous and speaks to the power of modern molecular medicine. However, much work needs to be done. Anti-VEGF therapy is, however powerful, only a symptomatic treatment. The underlying disease remains and is only kept in check by a lifetime of relatively frequent intravitreal injections. We need longer acting treatments. And as I said, some patients' disease doesn't progress, but they don't regain much vision. So we need a better understanding of what really is driving the pathology so we can intervene more effectively. And there remains no treatment at all for dry or atrophic AMD or geographic atrophy, which also inexorably causes vision loss and blindness, albeit with a slower time course. So our two speakers today have done and are doing significant work towards meeting the still unmet medical needs in AMD. Before I introduce our first speaker, I wanted to remind everyone that we will be taking questions at the end of the event after both speakers have completed their preparation. And you'll see a Q&A box at the bottom of your screen, which will be live throughout the entire event. So if you click on that, you'll have a box and you can type questions into the box, which will show up for all to see. So if you think of a question during the presentation, please type it into the box and we will do our best to get to the questions during the Q&A time. You'll also be able to add questions after both speakers have completed their presentation during the Q&A session. Okay, so let's get on to the main event. It's my pleasure to introduce Aparna Lakkaraju, PhD. Aparna is an associate professor of ophthalmology at UC San Francisco, and she runs a lab that performs research related to AMD and inherited diseases as well. Dr. Lakkaraju and a team have pioneered live cell imaging to identify functional defects in the retinal pigmented epithelium. And I believe we'll be hearing about that today. They've also extended that technique to the living retina. So she's working on helping to identify FDA-approved drugs that efficiently target disease pathways. She has had numerous awards and honors, including RPB Career Development Award, RPB Macular Degeneration Foundation Catalyst Award for innovative approaches to AMD. So without further ado, let me give you over to Dr. Lakkaraju.
Thank you very much, Carl, for that kind introduction. And thank you to Brian and RPB for the invitation to speak here today. So as Carl mentioned, my lab is interested in identifying mechanisms that drive vision loss in age-related macular degeneration, with the ultimate goal of using this information to develop effective therapies. And today I want to share with you some of our recent studies where we've used advanced live imaging to understand the mechanisms behind this disease and how this has helped us identify some promising therapies. So before I delve into the science, I want to acknowledge the bright, passionate, and conscientious people in my lab who perform most of the work. And I also want to take a minute to thank Research to Prevent Blindness that has generously supported my lab from the early days when I was a newly minted assistant professor with a Career Development Award, and more recently with the RPB American Macular Degeneration Foundation Catalyst Award for innovative approaches to AMD that has helped us build on some of our earlier discoveries and approach the problem of age-related macular degeneration in a more creative manner. So we know that age-related macular degeneration destroys central high-resolution vision. But perhaps we can truly understand the devastation caused by this disease when we look at the work of artists that have suffered from it. Here is a couple of paintings by Georgia O'Keeffe, and on the left is a painting that she painted when she was in her 40s. And this painting also has the distinction of being the most expensive piece of art by a woman artist ever sold. And on the right, you have a painting that she made when she was in her 80s, had advanced age-related macular degeneration and could no longer see. Now, while Georgia O'Keeffe had age-related macular degeneration, Edgar Degas had an inherited form of macular degeneration, what we now think was most likely Stargardt's disease. And he began losing vision fairly early on in his life. And you can see that a painting on the right here that he painted when he was in his 50s lacks many of the details that are very clearly visible from his work when he was in his 30s. So the macula is the part of the retina that senses that is responsible for high resolution vision. And this is the part that is destroyed in age-related macular degeneration. So those of us with healthy maculas and healthy vision can clearly see the faces of these two boys, but those with AMD see something like this. So these patients cannot read, write, see the faces of their loved ones or drive. And currently in the United States, 15 million people suffer from age-related macular degeneration. And globally, this is around 30 million. In the US alone, the number of people with age-related macular degeneration is greater than those with Alzheimer's disease, Parkinson's disease, breast and prostate cancer put together. And because of a lack of effective therapies, like Carl mentioned, the number of people with AMD is projected to go to up to 200 million by the year 2050. Now, why some of us age normally and hang on to healthy vision while others go on to develop age-related macular degeneration is still not clearly understood, but some combination of genes and environmental factors are thought to contribute to the disease. So before we go into the disease, I just wanted to take a minute to talk about how we see and where does macular degeneration begin. The eye is very much like a camera. So just like a camera, light enters the eye through the cornea, is focused by the lens, goes through this gel-like substance called the vitreous until it travels all the way back to the retina, where you have these photoreceptors, which are the light-sensing cells of the eye. These photoreceptors absorb, convert the light signal to an electrical signal, and then transmit it back to all the other layers of the retina through the optic nerve to the brain, and that is how we see. Now, because these photoreceptors are the first cells that sense light, they're constantly subjected to light damage and oxidative stress, and they constantly need to be taken care of, nourished, and supported. And this support is provided by this layer of cells called the retinal pigment epithelium, or the RPE. Now, this layer is the first site of damage in the most common form of macular degeneration called dry AMD. And this is also the tissue that we study in my lab. Now, like I mentioned, the retinal pigment epithelium sits beneath the photoreceptors and performs many functions that are absolutely indispensable for vision. And these include the absorption of vitamin A from the circulation to send it to the photoreceptor so that we can see. The RPE, the retinal pigment epithelium, has pigment granules, therefore the name, that absorb stray light and improve the quality of the image that we see. Perhaps one of the more important functions that the RPE performs is that every day, the minute we open our eyes, these photoreceptors, to account for the light damage that they go through, shed about 10% of their length that is then taken up and chewed up and digested by the underlying retinal pigment epithelium. Now, there are a few things to remember here. First is that just like the neurons in our brain, the photoreceptors and the retinal pigment epithelium do not divide. So we only have what we are born with for the most part. And now in the human eye, each retinal pigment epithelial cell takes care of about 30 to 40 photoreceptors. So you can imagine every day taking in 10% of 30 to 40 photoreceptors, how enormous a burden this is. This is like eating 100 bagels a day, every day, for the entire lifespan. So over a lifetime of eating these 100 bagels a day, what happens is that the RPE accumulates debris both within the cell in the form of lipofuscin, which are vitamin and metabolites, and above and beneath the cell, which called drusen, which are lipid protein aggregates. Both lipofuscin and drusen are associated with inherited and age-related macular degenerations. So how do we go from a healthy, happy retina where the RPE are performing its functions, taking care of the photoreceptors healthy and functional, to AMD? What happens is as we age, all of us start accumulating lipofuscin, these vitamin A metabolites that I mentioned. And as we further age, we do accumulate drusen, but in some cases, these drusen become larger and they begin to lift off the retinal pigment epithelium by putting stress on it. And because each retinal pigment epithelial cell takes care of about 30 to 40 photoreceptors, this causes a loss of the photoreceptors. Now, this loss of the RP and the photoreceptors due to drusen is called dry AMD or geographic atrophy, and it causes chronic vision loss in about 90% of the patients. And as Carl mentioned, we do not yet have any approved therapies for this disease. Now, when you have the blood vessels that are sitting beneath the retinal pigment epithelium that grow into the retina and lift it off, this causes what's called wet AMD. And this causes acute vision loss in about 10% of the patients and is currently treated with anti-VEGF therapy. And Dr. Green, who's the other speaker, will speak more about this. So one reason why treating dry AMD or finding effective therapies for dry AMD has been very difficult is that this is a very complex disease. There are about 50 independent genes that are associated with the disease, and these modulate many different pathways such as cholesterol regulation, inflammation, mitochondrial function, and debris clearance. However, not everyone who has these genetic polymorphisms goes on to develop dry AMD because an additional layer of complexity is conferred by other factors such as age and lifestyle choices such as smoking, diet, and exercise. So putting all of this information together and trying to identify therapies is almost like sitting with a 10,000-piece puzzle of the night sky, and all we have figured out so far is where the moon goes. So my lab has taken a different approach, and what we focus on is the retinal pigment epithelium because this is the primary site of damage in dry AMD. And because it's a very dynamic disease, we use advanced live imaging methods to follow real-time changes in healthy and diseased RPE to understand what is happening and where the cell tips between health to disease. And because multiple genes and environmental factors are associated with AMD, instead of targeting a single gene or a single environmental factor, we want to target biological pathways and identify therapies that can target these pathways. And finally, we want to put all this information together to understand how genes that are associated with AMD can drive the disease. So what we do in my lab is we begin with what we know from human data, from genetics, epidemiology, and from studies on donor retinas. And from this, we then go into our models in the lab. We use several different models. The in vitro model, we culture RP from pig eyes or human donors, and RP sheets in culture form these beautiful cobblestone structures that you see here. We also have mouse models of the disease. And we use these models for live imaging, and this live imaging techniques that we use can identify or follow events inside the cell in millisecond timescales and at nanometer resolution. And we use these imaging methodologies to discover disease mechanisms and potential drug targets. And then once we identify some mechanisms and targets, we go back into the human donors to confirm our data. So using these models, my lab has been interested in understanding what we like to call the 3 horsemen of AMD, which are increased debris, metabolic deficits, and chronic inflammation. And all these horsemen then lead to the 4th horseman, which is RP cell death and progressive vision loss in dry AMD. So what we noticed when we first began studying human donor tissue is that compared to normal donors, these are sections of the retinal pigment epithelium, the donors with AMD have a lot of debris shown here with in green, and also increased markers of inflammation also shown here in green. Now, the cell has many ways to cope with increased debris and inflammation, and it has specific compartments that perform specific functions in dealing with these. So these compartments need to be transported to particular locations in the cell. They need to meet and talk to one another to perform their functions. As an example, you have these structures or compartments called autophagosomes that you can think of as the garbage or recycling trucks that come to your house to collect the garbage. And then they have to take the garbage to the lysosomes or the disposal facility. Okay, and so this transport between the garbage trucks and the disposal facility is carried out by cellular highways called microtubules. these microtubules are very dynamic in the cell. They come together and come apart based on what needs to be transported at what part of the cell at which time. So you can see here in this really cool movie that these microtubules are coming apart and coming together. And when they need to transport cargo, for example, the garbage that is picked up from various parts of the cell, they walk on these microtubules, and that's how they transport this cargo. So when everything is going well and the microtubules are highly dynamic, the traffic is flowing free in the cell and the garbage is being disposed of and recycled perfectly fine. But when these microtubules are no longer dynamic, you have these static microtubules that lead to traffic jams within the cell. So what we found in my lab is using our models of macular degeneration. We found that in the RPE, in AMD tissues and models, there is increased accumulation of these two fatty molecules or lipids called cholesterol and ceramide. These two lipids then cause an increase of static microtubules that lead to traffic jams and prevent the disposal of garbage within the cell. So this is something like if the garbage disposal system in your sink is all clogged up, then you know that the garbage is not going anywhere. What we also found was that drugs that are currently approved to treat various conditions like antidepressants or osteoporosis can prevent this cascade and relieve the traffic jam in the cell. So what we asked was, will restoring the flow of traffic using FDA-approved drugs decrease the accumulation of debris within the retinal pigment epithelium? So what we did here is we tagged the garbage trucks, the garbage recycling trucks in green and performed live imaging within the retinal pigment epithelium. And you can see that in a normal healthy cell, these recycling trucks are moving just fine. And what the postdoctoral associate in my lab did was she tracked each of these trucks and she tracked how far they move, in which direction they move. And she found that in the healthy RP, everything was going just fine. But in RP that were modeling macular degeneration, what she found was that these trucks are not moving anywhere. They're just sitting around and they're accumulating the garbage. The garbage is sticking around. So when we then took these diseased RPE and treated them with the antidepressant, what we found was that we can completely restore the trafficking of these garbage and recycling trucks. They go on to be delivered to the disposal facility, and the debris is cleared in the RPE. So far we have this mechanism where we have in macular degeneration, the accumulation of these two lipids or fatty molecules that cause traffic jams that then leads to increased debris. And we also have a very promising drug candidate that can halt this cascade and lead to a proper clearance of the debris in the cell. Now, what about metabolic defects and chronic inflammation? To our surprise, what we found was that the same pathway also applies for these two horsemen in the RPE, only in this case, rather than traffic jams, what metabolic deficits and chronic inflammation are consequences of mail that is received and sent by the RPE. Now, to put this in a context, the RPE communicates with other parts of the retina, like the photoreceptors and the systemic circulation, to make sure that the retina is healthy and the nutrients get into the retina and metabolites get out of the retina. So on a very simple level, what we asked is, what is the RPE sending out and what is it bringing in? And here what we did was we tracked mail that is delivered to the RPE. And here my graduate student performed live imaging, and here the RPE cell is labeled in green and the mail that is coming in is in red. And you can see that in a healthy cell, the mail that is coming in is looks normal, all of the uniform size, and we didn't find any differences in that. But in the diseased RPE, you can see that you have these gigantic envelopes that are coming into the cell that the RPE is taken in. And what we found when we analyzed what the RPE is taking in, we realized that the mail that is being sent into the RPE disrupts metabolism and energy supply and also increases the propensity of these cells to become inflamed. And we also found out that in our mouse models, when we treat these mice with the antidepressant drugs, we can prevent the delivery of the harmful male to the retinal pigment epithelium.
Dr. Lakharaju, you're at the 18-minute mark if you could finish it.
Thank you, Brian. So now comes the big question, are people who take these antidepressants protected against age-related macular degeneration? And the simple answer is yes. This was a study performed about almost 2 decades ago now, where they found that people who took these antidepressants had a significantly lower incidence of developing age-related macular degeneration. So now my lab has a patent on this, and we are finishing up preclinical studies with the goal of starting clinical trials sometime in the future. So to just wrap up with some perspectives, the macula, you can think of it as the most valuable real estate in the body because the loss of function severely impacts the quality of life. Where we are, we understand the genetic basis of AMD. We have developed powerful non-invasive imaging techniques that can not only track the development of disease in people, but also track response to therapies. We have preventive approaches for dry AMD and therapeutic approaches for wet AMD. I'm hopeful that in the future, with deeper insight into the biology of dry AMD, we can develop effective therapies either by repositioning already existing drugs or by finding novel drug targets that can help preserve healthy vision over the human lifespan. So until then, here are a few lifestyle choices that we can all make. We can eat healthy, take antioxidants, exercise, quit smoking or don't smoke, and wear sunglasses. So with that, I'd again like to take a moment to thank all the bright young people in my lab, our collaborators, our funding sources from the NIH, Bright Focus Foundation, Macular Society UK, and of course the RPB and American Macular Degeneration Foundation Catalyst Award. Thank you.
Really excellent talk. Thank you so much, Aparna. I've got lots of questions. Now I'd like to introduce our next speaker. Our next speaker is Jordan Green. Jordan Green is professor of biomedical engineering and also ophthalmology, neurosurgery, oncology, material sciences and engineering, and chemical and biomolecular engineering at Johns Hopkins. That's quite a mouthful, Jordan. He is the director of biomedical engineering undergraduate program. He is the founding associate director of the Translational Tissue Engineering Center and the founding associate director of Translational Immunoengineering Center at Johns Hopkins. So Dr. Green wears lots of hats at Hopkins. I don't know how he can possibly do all those jobs at once, but that's terrific. So Dr. Green also is an entrepreneur. He was the founding CEO of the biotech company Asclepics Therapeutics. He currently serves as a member of their board of directors. He has received numerous awards, including the Presidential Early Career Award for Scientists and Engineers, Popular Science's Brilliant 10 Award. He is a National Academy of Medicine Emerging Leader, and RPB James and Carol Free Catalyst Award for Innovative Approaches in AMD. So Dr. Green and his lab are inventing the future of medicine through innovation of biomaterials, nanotechnology, and advanced therapeutics, especially working in ocular therapies as part of the Wilmer Eye Institute. And so without further ado, let's get to Dr. Green.
Thank you so much, Carl, for that kind introduction. And thank you so much to RPB and the James Carroll Free family for their support of our work. And thank you to Aparna for the great first talk to really set things up for what I want to talk about here, which is more on the applications and design of new types of therapeutics side to AMD research. And so what I'd like to share with you today is how we're using biomedical engineering approaches to develop new types of technologies to treat wet AMD. And as a disclosure, I am a co-founder and equity holder in the company Asclepics Therapeutics and a member of the board. And then have also developed different patents related to technologies at Asclepics and some other technologies that then are in this presentation. And any potential conflicts of interest are managed by the Johns Hopkins Committee on Outside Interests. And so the the challenge that we've heard with AMD is that there needs to be improved therapies. And so on the one hand, we're looking for new types of approaches that can have new mechanisms that could enable greater potency, broader activity, new types of mechanisms in terms of looking at both VEGF, but looking at other types of mechanisms too, such as TIE2, and I'll talk a little bit about this. And then in addition to improved efficacy, we're also looking for long-lasting durability. So even though many patients are being managed with their care with wet AMD, it involves frequent injections, right into the eye and into the vitreous. And so wouldn't it be better if there was just one injection that was needed every 6 months or one injection every year? And so this is something that we're very interested in, is improving the durability. And then finally, to also look at approaches that could have broader accessibility. So other kinds of technologies that maybe are less expensive to manufacture or easier to administer to help patients further. So those are the challenges. And so with the first approach, what we looked at was a new type of peptide to treat wet AMD. And where this peptide comes from, it's a natural amino acid, and it comes from collagen. And so if you think about cartilage, you don't have blood vessels growing in your cartilage. And it's in part because there's these natural molecules that then prevent that. And so what we wanted to do was to look for natural molecules, these peptides, that are able to maintain homeostasis with how blood vessels grow and to prevent abnormal blood vessel growth. And so that was the roots of this peptide as a drug. And so how it works is it's able to actually turn off the signaling of VEGF, which is the well-known clinical target. And it does this by degrading the receptor that then that VEGF binds to. And I'll have a slide where I'll talk about that a little bit. And in addition, the same peptide also activates TIE2, which is another clinically validated target, which is important in having strong junctional integrity between the endothelial cells and preventing leakage. And so I'll go through that in a few slides. But the concept here is that we'd have a new type of agent. Instead of having a large protein or an antibody, it's a small peptide, but that it could have enhanced efficacy potentially, as well as long-lasting durability, and then be easier to manufacture. And so that's what we wanted to do. This is what the peptide looks like. It's a 20-mer. So this is pretty small, and it kind of curls back on itself like this. And so this is the peptide AXT107. It's a biomimetic peptide that is very similar to a sequence that naturally occurs, again, in collagen, and that it's able to restore vascular homeostasis and prevent angiogenesis. And so this is a little bit busy, but the key point here is just to show that the peptide, the way it acts is it acts on integrins, and it disrupts the integrins. And so what this does is it then leads to uptake and degradation of this receptor, VEGFR2, which is very important because VEGF binds to this receptor, and this is the signaling that leads to a lot of angiogenesis and leakage. And so by turning off the receptor, it's turning off the ability of VEGF to signal. And then in addition, the same peptide, by disrupting integrins, also causes clustering and activation of TIE2. And this is important to tie the endothelial cells together and prevent leakage. And so this is important for wet AMD. It's important for diabetic macular edema. And so this is the strategy with how the peptide works. And so we did different studies, and these were initially in mice and rabbits, and in collaboration with Peter Campochiaro at Johns Hopkins and the Wilmer Eye Institute. And so what we found in different models was good efficacy. And so we initially looked at VEGF-based models. So this was a genetic model where there's a lot of expression of VEGF that leads to leakage and even retinal detachment. So it's very severe, the disease. And so in the control case, all of the eyes have retinal detachment. This is in mice. And then what we found is with leading anti-VEGF agents, it significantly reduces, but there's still a little bit of total detachment. And then with AXT107 at a much smaller dose by mass, we're able to stop all of those total detachments, even this very strong VEGF-based genetic model. Then we looked at a model in the rabbit eye, and this is using a bolus VEGF to create leakage. And then we see the peptide's able to reduce that leakage, and then it persists over time and compares favorably to leading anti-VEGF agents, and that even after 2 months, you'd still have very good control of the leakage, where anti-VEGF could be stopping working at that point. And so this led us to see that it was a very promising candidate to move forward with. But then when challenged is as a peptide, it could potentially be degraded and cleared really quickly. And so we wanted to be able to enable long-lasting durability because, again, you wouldn't want frequent injections in the eye. And so our first approach was to use particles that we made different, using an engineering approach. We made different biodegradable particles that could safely degrade in water. And then we would put the peptide inside of them. This is what they look like. One of the things we found is if they had an elongated shape, they actually persisted longer and didn't have the same kind of immune response, weren't being able to be eaten by different kinds of immune cells. And so this was helpful. And that they could then release the peptide over time. You could have many months of release from the particles. And then when we looked at their efficacy, the amount of green here is the amount of neovascularization. So we want to stop that. And so here's an empty particle, here's the particle with the peptide, that it does reduce that neovascularization. So it did look like these particles could work, but they do introduce another complexity of making the particles, and then the length. Here we have about 4 months, but we wanted to look at longer time points. So then we were thinking, are there other approaches we could use to extend the durability? And the idea here was to make a gel depot so that the injection would lead to a self-assembled gel that would just sit below the visual axis. It doesn't interfere with vision at all, but that it could slowly release as a gel. And so this is what we then worked on, and we found these peptide gels could enable slow release, and that if we looked at, for example, leakage in the animal models, this is in rabbit, that we see that with there's a dose response, with the higher dose, especially this long persistence of reduced leakage. And then the visual axis is clear. There's no interference with that gel with vision. It just sits at the bottom of the eye, and then as this depot, and it slowly dissipates over time. So that's what we wanted to look for. And so we felt things were very successful with this project. We're able to show sustained delivery of the peptide through 2 different systems, through the particle or through the gel. And in both cases, it's efficacious and long lasting. And then we validated this in different animal models, and we looked at the biodistribution, and we looked at the safety, and everything looked really good. And so where this has moved forward is that Asclepics actually has raised money to start clinical trials. And in December 2020, the FDA approved the IND, and that now it's being administered in patients for different types of retinal diseases. And so we're interested in neovascular AMD and diabetic macular edema, and also edema following retinal vein occlusion. And things are going well, and the formulations, and when they're injected in patient eyes, it looks the same as it did in those preclinical animal models of that depot of the gel, but then slowly dissipates over time. And then we were very interested in looking at other technologies for wet AMD as well. So then as this has been successful, we've also pivoted to other things that we could engineer as biomedical engineers. And here we're looking about, well, what about if we want to turn on or off other types of factors? And as we heard in the first talk, with dry AMD and with other retinal diseases, there's lots of different proteins that could be at work. And so if we could modulate those on a genetic level with a technology that was very robust, this would be really helpful. And so could we deliver nucleic acids that could be then expressed as proteins and have very long-lasting durability, maybe even lifetime with 1 dose? And could it be done in a safe way? And so there's a lot of work using viruses for gene therapy, but what we wanted to do is to use a similar approach as before of using biodegradable materials without any viruses. And so this is a biodegradable plastic that can form a nanoparticle that can then deliver the cargo, these genetic cargoes, to then turn on genes, have long-term protein expression in the eye. And so the challenge of intracellular delivery, and this is a little bit like the first talk, except in this case, it's not garbage, it's very important, precious cargo that we want to deliver into the cells. And so how we start is the biodegradable polymer binds the nucleic acid, the DNA, encapsulates it into this particle that then safely and effectively gets internalized in the cells, into these endosomes, and then has a mechanism to get out of the endosome into the cytosol. And this is the main part of the cell. And then here it degrades safely in water releases the cargo that can then make its way in the nucleus and be expressed. And so this is what we wanted to design. And the way we designed it is we first looked at different polymers. In our lab, we designed new types of materials, and including polymers, which are plastics. But then these are plastics designed to be biodegradable and safe. And that we designed them to be able to bind DNA and enable these different steps in delivery to get taken up by cells and get out of the cytosol out of the endosome into the cytosol and then degrade in the cytosol. And so that's what we built into the material. And so this is what the particles look like. They're about 100 nanometers in size. This is really small, about a thousandth the width of a human hair. And then what we're also interested in is how we're going to inject it in the eye. So instead of injecting it right into the vitreous, like we did with the self-assembled gel, what we're doing instead is this suprachoroidal space. And so the sclera is the white part of your eye, and then the choroid, this is where the blood vessels are. And so just in between the sclera and the choroid, there's this potential space. And so we can inject in here, and then the volume in these particles can spread out all around the back of the eye. And so that's what we wanted to do. And so what we saw is widespread expression. We initially did a DNA that encoded a green fluorescent protein. I don't know if some of you have been to an aquarium and seen jellyfish that glow green, but it's that same gene, just as a marker to see if this works. And we did see very good expression, all through the retina. And this is something that we looked at, that spatially we see this expression all the way through behind the retina and around the other side. So we have this good distribution of the gene delivery. And then we also see really good persistence. So with one injection, we see that this fluorescence lasts a long time, 6 months. The longest we looked out to is 8 months, and we saw very persistent gene expression in the retina and choroid, showing that this delivery material worked really well. And that if we wanted to do multiple injections, unlike with a virus where that's difficult due to immune responses, we could do repeated injections. And as we do more injections, we get higher levels. And so this looked, really promising to us. And just to look at the retina itself, if you look at it like a flat mount, you can see, again, everywhere you see a green signal, that means that nanoparticles successfully went through the cell, got out of the endosome, degraded, released the DNA made its way in the nucleus, got expressed to mRNA, got translated to protein, and then you see the protein. So all those steps had to happen to see a little green dot. And what we see is all through the retina, we see lots of green dots of this happening, all through the retina. Or if we look from the other direction, we see this, really strong fluorescence. And so instead of just looking at fluorescence, we wanted to say, well, could this be functional? Could we deliver other genes and to modulate the disease or create models? And so the first thing we wanted to do was to create models for wet AMD so that there could be new ways to study it. And what causes this phenotype in these models is overexpression of VEGF. So we wanted to deliver in our nanoparticle a gene for VEGF. If it worked and VEGF was expressed, what we would see is new vasculature growing, and we'd see leakage. And so that's exactly what we saw. So we do one injection of the nanoparticles, and then the animals start developing wet AMD in a way that really matches what one would see in patients, including that there's this progression of subretinal fibrosis. And so it really looks like wet AMD, but it's done with just a simple injection. And now we can look at these animal models as ways to look at new treatments for the disease. Now, we don't want to just cause a disease. We also want to look if we can treat it. And so the idea here is we had genetically encoded an anti-VEGF agent. So we could do a different animal model and then do one injection of the particles to see they express the anti-VEGF agent. Does this help treat wet AMD? And so this is sFlt1, and so we have good levels of the protein that get expressed. It reduces leakage that we measure with albumin, and then it also reduces neovascularization. That all the red you're seeing here is the new vasculature growing. This is wet AMD happening, and then with the nanoparticle treatment, this has been reduced. So what this shows then is proof of principle that in a non-viral, biodegradable plastic particle, really, that we can safely turn on a gene of interest. And this is something that we can just mix in and out different genes or to probe different mechanisms to see what would be good to deliver. And then unlike making protein, recombinant protein, this would be an off-the-shelf particle that delivers the gene, and then it could have this very long-lasting efficacy, 8 months or longer, maybe years following the one injection. And so we're definitely interested in continuing this work. And so the future directions with the peptide, it's now being evaluated in patients with wet AMD and DME. And on the non-viral gene therapy, we're doing the fundamental research on trying to further improve the nanoparticles, the suprachoroidal injection methods, and then the types of genes that we want to deliver for efficacy. And so with that, I like to thank you so much for your attention. I'd like to thank again RPB and the Free family for all their support. And I'd be happy to answer any questions. And this is my lab, and very thankful to them as well. Thank you.
Thank you very much, Jordan. Terrific talk. Again, lots of interesting things. Lots of interesting things. So so let me start the discussion here. Let me start with a question for Aparna. So Aparna, in your talk, you showed some really cool images of subcellular particles moving around. And I was wondering, you also mentioned that you did live imaging. Were those videos all taken from cells, or were some of them from living animals?
So these videos are from retinal pigment epithelial cells from pig eyes, but we do have images of movies where we can track mitochondria and calcium imaging and all of that in the living retina.
That's remarkable. What kind of optics do you use?
So we are using a spinning disk microscope with the latest technology, with a magnifier and also a chamber to maintain the temperature and the carbon dioxide and everything, and also at a very, very low light level to not cause phototoxicity.
Let me once again remind everybody in the audience to please feel free to ask questions, put them in the Q&A box. In your cells, another question, in your cells, you spoke about the diseased cells. So how do you mimic AMD in your cultured RPE cells?
So we do two things, because we want to look at the earliest stages of dysfunction, because what we think is that if you can stop it at the earliest stages, that gives us the most chance of protecting healthy vision. So we have cells with lipofuscin, which we all accumulate as we grow older, and has been associated with inherited macular degeneration. And then we challenge the cells with complement, because complement activation is one of the biggest risk factors for AMD. So what we find is some of the data I showed today, some of the movies, the autophagic defects or the garbage disposal defects, those are found even when the cells accumulate just lipofuscin. And then when you add complement, what we showed was that this causes mitochondrial fragmentation and oxidative stress.
Now complement can actually kill cells. I mean, you can get completely broken membranes if you have
Yes.
the MAC complex. So you're activating complement in some nuanced, subtle way.
Yes. We are doing a sublytic dose where the cells are just, because this is again, like I said, we want to identify mechanisms at a very early stage, not where the cells are already dead and are dying. So we're doing it at a very low level of complement activation.
Fascinating. I have one more question. I have to take the privilege of the moderator to ask my questions. The tricyclic antidepressants that you mentioned, they're old drugs, and they are traditionally quite dirty drugs. They interact with multiple transmitter receptors. They interact with multiple transporters. Do you have any idea what receptor they're going and interacting with?
So what we're looking at is this enzyme called acid sphingomyelinase that these drugs inhibit as an off-target effect. And this has been shown for many years in the neuroscience field, actually, that these tricyclics and even SSRIs inhibit this enzyme through an off-target effect. And that decreases the lipid ceramide, which then restores trafficking and protects the mitochondria. So it's completely separate from the central effects. And in the mice, the doses we use are about 1/5 of what's required for human beings to achieve central effects.
Very good. Okay, so I see we have some Q&A, some questions in the Q&A box. Let me see that. And they're not particularly directed to Aparna or to Jordan, so I'm going to throw it out to both of you. Can you discuss the likelihood that dry AMD treatments that interfere with the complement pathway, such as a pegcetacoplan, might have, I presume they're asking, do you think that those are promising therapies?
So I think the question with COVID-19 is that there's these recent papers that came out showing that AMD patients have increased morbidity and increased, they need more ventilator support when they get COVID-19, and because complement also plays a role in COVID. But with this particular drug, with Apellis, I think from the latest studies that we've seen, and Carl probably knows more about this, is that it also causes an increase in wet AMD. About 20% of the patients develop wet AMD. So I'm not entirely sure how that's going to go with regards to FDA approval.
Interesting. Here's another question, and perhaps Jordan can take this one. It says, can you discuss the possibility of topical transscleral delivery of treatments? Have you thought about that, Jordan?
Yeah, so that's a great point. So there are a lot of other approaches to the administration of these drugs, right? And so you could think of systemic IV administering, intravitreal, the suprachoroidal, and then there's a topical. So with topical, the challenge is that in some ways it's easier to, if someone could do their own drops, let's say, but then a lot of the drug just gets cleared really easily from blinking and from mucus. And so it's very hard to get the drug, especially to the back of the eye. Now, there's some companies that are looking at this with nanoparticles or other kinds of formulations. And so it's a small amount of the administered drug, but that it could reach the back of the eye. So this could be an approach for the future. The challenge is that the patient would have to repeatedly use these drops. So maybe daily or even multiple times a day. And so, you know, is that ideal, or would having an injection that could last 6 months or a year maybe be better with compliance? But it is potentially possible to formulate medicines that with the drop, a small amount could reach the back of the eye, but it's challenging. With other kinds of routes, transscleral, yeah, you could have different kinds of depots that then are put in other places where you could have slow release and diffusion to get to the back of the eye too. And so these are methods that are in development. And I don't think there's necessarily a best mode of administration across the board, but I think looking at these other modes of administration could enable certain kinds of medicines to have really good durability or easier you know, application.
Interesting. Okay, so another question here for Dr. Lakharaju. Is it known that lipid ceramide accumulates in the RPE cells of aged human RPE or mouse models?
Yes, so we have published data showing that in the mouse models, there is increased ceramide in the RPE. And we are just, hopefully this paper will come out soon, but we have also data showing that in AMD donor RPE, there is significantly more ceramide compared to unaffected donors. And in fact, there was a study out of Germany that was published a few years ago showing that AMD patients have more ceramide in their serum compared to unaffected patients. So there's definitely, it could be something linked to the serum ceramide or ceramide in the RPE, but yeah, we do have data on that.
Okay, I have a question for Dr. Green. So a couple of questions. One is concerning your gel delivery system in the eye. How does it, in the intravitreous for your first drug, how does that erode? Does it fragment? Does it dissolve in? Does it become full of liquid and dissolve all the way through like a sponge? Does it fragment? Can you describe the solution of the gel?
Yeah, so the way it works is that it's something where the peptide is fully soluble in water when it's a lower pH, but then when it's at neutral pH and in the environment of the vitreous, it then self-assembles quickly into this gel. And so there's attractive forces with the peptide to associate with itself non-covalently, just
I see.
physically.
It actually isn't a special gel. It's the active principle itself.
That's right. It's not a separate biomaterial component. It's the peptide itself. A few of the amino acid residues are different than a naturally occurring
I see.
peptide it was based on, and so that was done to help with a better KD to its
So
target.
you know, when I put something into a media and it forms a solid, I usually say that means it's insoluble, and that I don't really have control over what's actually forming the nature of the gel and the kinetics of release. Have you done kinetic studies to show
Yes.
that it's released in some sort of steady fashion?
That's right. So it's something where it's not forming a solid, it's forming a gel. And then we've done kinetic studies, careful kinetic studies in different animal systems and in also human vitreous. And what we see is that about half of the material remains after 6 months, and that this is consistent across a wide range of doses, so that it's got very consistent kinetics of an exponential curve where the half-life is 6 months. And so if you start with more, it's just you'll have more longer, but it follows the same disassembly. And what it looks like is the depot just gets sort of lighter in density and smaller, and that it just keeps growing smaller and looks more diffuse over time. And so this is what made it attractive to not have to use then a separate biomaterial-based system in addition, but that just the drug itself was designed that it would have this property.
Interesting. Okay, a few more questions from the chat here. I guess this is for Dr. Raja. Will antidepressant use stop an already occurring macular degeneration, or is it only good to prevent the disease?
Well, we are hopeful that it'll slow the progression of the disease. So like I said, we still need to do studies in humans. That's the gold standard, of course. But in mice, at least, we begin treating them at 6 and 12 months after the disease has begun. And we do see improvement in the health of the RPE and the retina. So we are hopeful that we will see similar things in humans too.
Maybe anybody can answer this question, because I don't think that it was discussed really in either of your talks, and there may be a typo in this question, so I'm going to make a guess. But can the panel discuss effects, if any, of chemotherapy on what I'm guessing is the progression of AMD? I have no, I actually, I mean, it's an interesting question. There's always drug interactions. You know, chemotherapy, classical chemotherapy with this, you know, rather toxic substances, cytotoxic substances used as drugs. You know, there are more modern chemotherapies now that are directed at cytokine pathways or that are used to boost the immune system, the checkpoint inhibitors, et cetera. So but I don't really know. I think that those drugs have many, many side effects, and I wouldn't be surprised if they have effects on AMD.
One role is some of the newer chemotherapies that are based on receptor tyrosine kinase inhibition could potentially then block the activity of VEGF, which is, you know, one of the main effectors with progression with wet AMD. So there could potentially be some benefit there, but again, it's something that's affecting many parts of different systems, and so it's not the ideal therapy.
Right. Well, it's 1:59. So Brian, Diane, do you think we should wrap it up?
I want to thank again our two speakers, Dr. Lakkaraju and Dr. Green. I feel like a little bit like science fiction come to life. So thank you, very exciting, very innovative. I also want to thank our funding partners and the donors who have made it possible for us to bring this research into being. Also, Dr. Romano, thank you for a great job of being the moderator. And all who asked questions and all who joined in today, I would ask our two speakers to just stay on for a couple of minutes. We have a few other questions about some diet and lifestyle issues that I'd like to get to, but I want to bring it to a close. I also want to note that our next Lunch and Learn will focus on diabetic retinopathy. It will be held on Monday, March 22nd. And this is the end of our session, the formal end, but I will just ask, be the prerogative of the host to ask Dr. Lakkaraju or Dr. Green, does a poor diet in terms of poor choices of fat or high glycemic index foods cause enough inflammation to initiate AMD? Any thoughts about that?
So there have been a couple of studies showing that people with diabetes actually are protected against age-related macular degeneration. I'm not entirely sure about the specifics of the study. But in terms of high fat, one other confounding variable with AMD is the fact that high HDL, which is the good cholesterol, is associated with increased incidence of dry AMD. So again, that is something that people are working out. In terms of diet, I think what's really important is to eat what Michael Pollan says, foods of all colors, right? And so things that are rich in beta carotenoids and antioxidants and all of that. And I think he also says eat something like eat good food, but not too much. So I think those are the two good tenets to have in terms of diet.
Great. Another question that just came in is, what antidepressant are you referring to that was used in mice?
So we used a couple. We used, like what Carl mentioned, the old school ones, desipramine, which is a tricyclic antidepressant, and also amitriptyline, which is used these days to treat as a prophylactic to treat migraines. We've also used SSRIs, Prozac, and the other one whose name I'm, so sertraline and fluoxetine. And we also have some very promising data on drug to treat osteoporosis, which is more a direct inhibitor of this enzyme that we're targeting. So that is, and we're using that at a dose that is about 100 to 500-fold less than the antidepressant. So we're hoping that that is the drug that we will be focusing on more in the future.
Thank you. And a scientist who is watching the presentation, we have many lay people, but also some scientists who are in the audience today, but a scientist asked that said, outstanding speakers, and would these presentations be available to share with my lab team? And we will send a link to everyone. We're recording the session and we'll put it on our website, and we'll send a link to everyone who has joined in today, including those who have been involved as presenters and the moderator. Again, I'll just thank you. I think that's the end of our questions, end of our session, but a great job by all, and thank you for those who joined in. Good day.